All right, everybody. Thank you again for attending. Welcome again to this next edition, actually the fourth edition of another biology EOC review. We're going to be talking today about genetics, specifically Mendelian and molecular genetics. First of all, let's talk about Mendelian genetics. It's called Mendelian genetics because of Gregor Mendel, the person who really came up with all this by cross-pollinating pea plants. And he was able to make these theories based on that. And this is some of the terminologies that we need to know. First of all, you got to know the difference between phenotypes and genotypes. Genotypes are simply the letters. You know, they could be big R, little r, or little r, little r. Phenotypes are the appearance. So genotypes code for the phenotypes. The genotypes are the letters. Phenotypes is what the letters represent. So big R, little r would be rough. Or little r would be smooth. The genotypes are the letters, phenotypes is the appearance. Homozygous versus heterozygous. Well, the prefix HOMO means the same. And the prefix hetero means different. So anytime you see the words homozygous or pure, that's always two of the same letters. So big R, or little r. If you see heterozygous, that means different. So the only time you'll see heterozygous or hybrid is if they're talking about the heterozygous genotype, which is big R, little r in this scenario. Oh, question for you guys. Using the letter R, type in the chat box which two genotypes would be used when crossing a heterozygous round versus a wrinkled plant. What do you think it would be? Heterozygous round versus wrinkled. So which two genotypes are you crossing? Give you guys about 10 more seconds. Got a couple mixed responses here. Let's see which one's correct. All right. So when we're crossing a heterozygous round, remember, hetero means different, right? So we got a big R and a little r. Versus wrinkled. Well, if heterozygous is round, that means round is dominant all the time. So wrinkled is going to be the two little r's. Does everybody see how I got that? When you're crossing a heterozygous round, big R, little r. With a wrinkled plant, the only way something can be wrinkled is the recessive, so that's little r, little r. So Catlin, good job. You fixed it before we went over that question. But everybody else, just make sure that you know heterozygous is the big letter, small letter. Homozygous will be two of the same letters. Okay, so then you're going to cross them up. So we have big R, little r with a little r, which is the correct answer from before. The second step, so after figuring out what you're crossing, the second step is then you cross it. So you put a big R, little r on top. That's one parent. And we have a little r on the left. That's the second parent. It does not matter what parent you put on either side. But then you cross them up. And we get two big R, little r's and two little r's. So the chances of being round, that's 50%. 2 out of 4. Wrinkled, also 50%, because you got two little r's, or 2 out of 4. So your phenotype ratio, phenotype remember you're comparing what they look like. You got 2 round and you got 2 wrinkled. Very simple. If they ask you for the genotype ratio, it's also very simple as long as you know what you're doing. I got 0 big R, big R. I got 2 big R, little r's. And I got 2 little r, little r's. Everybody see how I got that? Once you cross them up, then you have to interpret the results on a Punnett square. So I got my phenotype, I got 2 round, 2 wrinkled. For my genotype, I got 0 big R, big R's, 2 big R, little r's, and 2 little r, little r's. So that's how you count and determine and analyze your Punnett squares. So step 1, you figure out what you're crossing. Step 2, you cross them. Step 3, you figure out the results and be able to tell me the phenotype and genotype ratio. Okay. Here's some more simple genetics. Gregor Mendel worked with pea plants to learn the basic patterns of inheritance. Phenotype, we already went over this, but phenotype is what the organism looks like, and the genotype are the letters or gene combinations. Homozygous, 2 of the same letters. Heterozygous, 2 different letters, but the same overall letter. Monohybrid cross goes through 1 trait at a time. And it looks like, you know, the Punnett square we just did. So here's an example of a monohybrid cross. And here is an example as well. So let's take a look at this first example of the monohybrid cross. Can anybody tell me what the phenotype ratio would be? And what the genotype ratio would be? So I need you guys to type in the chat box what you think the phenotype and genotype ratios are for this cross that has already been done for us. So I'll give you guys 30 seconds to figure that out. Okay, about 10 more seconds. Okay, so we're assuming that the big T stands for tall and the little t stands for short. So phenotype ratio, we got 4 tall and we got 0 short. So that would be correct, 4 to 0. Genotype ratio, if we look at our 4 results, I don't have any big T's. But I have 4 big T, little t's. And I don't have any little t's. So that would be your correct one. So good job, everybody who typed that answer. Phenotype is 4 to 0, 4 dominant, 0 recessive. 4 tall to 0 short. And genotype is 0, 4, 0, which simply means that there's 0 big T's, 4 big T, little t's, and 0 little t's. So I want you to do that one more time for this example here. It's already done for you. I just need you to analyze the results. So what would be your phenotype ratio? And what would be your genotype ratio? One more time. I don't know. I haven't changed my my tone of voice, but I'm sorry that my voice is a little bit off. Okay. All right, so if you heard, if you guys can put the phenotype and genotype ratio for the example right here, right above it. And it's okay to have mixed results. We just got to get the same results when we take the test. All right, so let's see how we did. We got big T, big T, big T, little t, big T, little t, little t, little t. So we have 3 dominant, which are the big T, and the 2 big T, little t's. And we got 1 recessive, little t, little t. So we got 3 tall, 1 short. It actually says it right here. Genotypes, though, let's check it out. I got 1 big T, big T. I got 2 big T, little t's. And I got 1 little t, little t. So it should be 1 to 2 to 1. So good job, Alana, for fixing that. Everybody else was close, but let me give you a little tip. Every time you add the numbers in phenotype and genotype, it'll always equal 4. Look, 4 plus 0 is 4. 0 plus 4 plus 0 is 4. 3 plus 1 is 4. 1 plus 2 plus 1 is 4. So every single ratio will always add up to 4. It's just how those numbers are spread out depends on what you're looking at. So that's how to do genetics, guys. That's a good chunk of your test. If you can, first of all, figure out what you're crossing, like seeing a question like this and coming up with these, that's the first step. You need to do that. Second step, if you know how to cross them. And third step, you know how to interpret the results that we just practiced with. Everything should be good to go in terms of genetics. In terms of percent, how much you think is genetics related? I'd say about 20% of the test is genetics, straight up genetics. So definitely you got to know, homozygous, heterozygous. You need to know dominant, recessive, things like that. Now for this question, you may see 1 or 2 questions on the test about it. This is called a dihybrid cross. The only difference is DI means 2, so it follows 2 traits. So instead of 4 boxes, it's 16 boxes. And I will show you guys briefly how to do this, but I want you to focus most of it on the simple genetics we just did. But when you do a dihybrid cross, all you're doing is just keeping 2 traits separate, but you're combining them at the end. So in this case scenario, they're crossing big R, big Y with little r, little y, little y. And to get the 4 different genotypes to cross, it's very simple. You just do the FOIL method. F means first. So what's the first 2 letters? Big R, big Y. O is the outer letters, so that would be big R, big Y. I is the inner letters, so that's again big R, big Y. And L is the last 2 letters, again big R, big Y. And that's how you get your 4 results. You just see the different combinations you can get from this genotype here. Same thing here. Little r, little y, little y. You figure your FOIL, first, outer, inner, last letters. And then you get your 4 different terms. So this one was based off of that. Right. I was showing you guys the parental generations, but for this specific cross, they're crossing the heterozygous of each. So for this one, first would be big R, big Y. Outer would be little r, or sorry, outer would be big R, little y, that you see right here. Inner would be little r, big Y. And then last would be little r, little y. So that's how you get your 4 different genotypes. And then the heterozygous up top, that's the same thing. So you've got to copy the same 4 again. And then it's very easy. That's the hardest part of the dihybrid cross. Figuring out these right here and figuring out these right here. But if you do the FOIL method, like you learned in math class, it does connect. You just figure out the FOIL method to get your 4 different genotypes to cross. After that, you just combine the letters, and it's very simple then. Genotypes and phenotypes will always add up to 16, just like we did from before, always adding up to 4. So again, I want you to know how to do this, especially how to set up the dihybrid cross with the FOIL method, but I want you to focus most of your time on the simple genetics that we just went over. All right, one other thing that you might see 1 or 2 questions on are pedigrees. Pedigrees are not difficult either, especially once they give you a multiple choice answer. So a pedigree is just a chart tracking 1 trait at a time. Square is male, circle is female. If it's not shaded, they don't have the trait. If it's halfway shaded, they are a carrier of the trait. So no trait may be little r, little r. Halfway shaded may be big R, little r. Shaded, that means they have the trait. So that would definitely be big R, big R. That's just a general key on how to use it. So looking at this pedigree here, it says which 2 individuals have the trait and got it from their father. So you have to tell me what row it's in, either 1, 2, 3, or 4, and then what number matches up with it. So I want you to look at this pedigree, and I want you to try to think which 2 individuals have the trait and got it from their father. Remember, a father is a square. So which 2 individuals have the trait and got it from their father? So think about it and try to type those in the bottom. Catlin, you got 1 of them right. I won't tell you which one, though. And you want to type it like, for example, this person right here, which is not the right answer. This one would be written I-1, Roman numeral 1, row I, number 1. So that's how you type it. Good job. Talea, you got it. Catlin, you got it. Here's an individual that has the trait that got it from their father. And here's a person that has the trait that got it from their father. The Roman numerals are just the different generations. Generation 1, 2, 3, and 4. Row 1, 2, 3, and 4. So your 2 answers should be, just like you said, 3-1. So it should be 3-1 and 4-3. So that's how to read a pedigree, guys. It's not too difficult. Just as long as you know the key, it's just a process of elimination. Look at the 4 answers they give you and see which one makes the most sense on the correct answer. Then you'll figure it out. And another 1 or 2 questions that you might see in genetics, this is complex genetics. These are genetics that don't follow the normal rules. Incomplete dominance is an example of that. That's where you get, normally it would be big R, red, big R, little r, also red. But in terms of in terms of incomplete dominance, this is where the heterozygous is mixed. So instead of red or white, so big R is going to be red. Little r is white. Well, in incomplete dominance, red or big R, little r is actually going to equal pink. There are 3 different phenotypes for incomplete dominance. So be careful. If they say incomplete dominance, that means the heterozygous is the blend between the 2. They write it differently, because they're all capitals, just because the prime just means it's just as strong as the other one. So little r, and R prime are the same thing. I'm just trying to show you where the whole kind of scenario works in. So if you see the word incomplete dominance, just know that the middle, or the heterozygous one, is a mix between the 2. That's different than codominance. In codominance, let's say big R is still red. We'll say little r is still white. Well, if it was incomplete, it'd be pink. But if it's codominance, big R, little r would be red and white. So there's your difference. Incomplete dominance, big R is red, little r is white, but the mix, the heterozygous, is pink in between the 2. If it's codominance, think of both. Codominance, big R red, little r white. Big R, little r is red and white, almost like half and half. So do you see the difference between incomplete and codominance? This doesn't happen all the time, but if you ever see these words, incomplete means pink, or the blend in the middle, and codominance means they all show up as the heterozygous. So know about that. Multiple alleles just means instead of 2 alleles that we practice all the time, like big R, little r, blood types are multiple alleles. They have 3, A, B, and O. So that's just an example of something with multiple alleles. Also, sex-linked traits is another example of something that is complex genetics. All that means is that it's going to be on, well, anytime you do a sex-linked trait, you always fill out the male and female first. Male is X, Y. Female is X, X. After that, then you fill out the genetics behind it. And in this case scenario, they're using a big N and a little n, and a big N, and the Y does not carry anything. And when you do the cross, you get 2 big N's here, you get a big N, little n here, you get a big N and a little n there. That's just crossing it up. The main thing you guys need to know with sex-linked traits, step 1, you put down the male, XY, and female, XX, on either end. Then you fill out the X's of whether it's homozygous or heterozygous. Big N, little n would be heterozygous. And then for the male, they only have 1 allele, so they will have the capital or the recessive allele. And then you just cross them up. This person will have the trait. This person's heterozygous, so they'll probably have the trait. This person only has the trait because they have 1 letter only. This person does not have the trait. So that's how you do sex-linked traits. Polygenic inheritance are traits controlled by many genes, like height. You don't get your height from just your parents' height. You get your height from many other genes together. Same thing with hair color as well. And then aneuploidy is a condition caused by having abnormal chromosome numbers. Like if you ever known someone who has Down syndrome, instead of 46 chromosomes, they have an additional chromosome. So they would have 47 chromosomes because they have a third extra chromosome on pair 21. So just know the basics of the complex genetics. You might see 1 or 2 questions about these, for sure. But again, I want you to focus most of your time on the Mendelian genetics, but you should also be aware of incomplete dominance, codominance, dihybrid crosses, sex-linked traits, polygenic inheritance. That just means traits controlled by more than one gene, or what aneuploidy is. That's just somebody who has an extra chromosome. And good question, Catlin. Person who has aneuploidy, they have at least a 50-50 chance of passing down that aneuploidy trait to the next generation. So as geneticists say, they make them aware of that, but it is a 50-50 chance, just like you do a Punnett square on it. So they could still reproduce, and it's a 50-50 chance of passing down that extra chromosome or not. All right, so here's a good question to work with together. Hemophilia, that's a sex-linked recessive trait. Which of the following describes the probability of hemophilia in the offspring of a man who does not have hemophilia and a woman who is a heterozygous carrier? All right, so I'm going to turn on the polling here. So if you guys want to choose A, B, C, or D, I will get you started. Remember, X, Y, here's male. X, X, here's female. So which of the following describes the probability of hemophilia in the offspring of a man who does not have hemophilia? So this would be not having it. So we'll put a little n. Remember, Y is nothing. And a woman who is a heterozygous carrier. So she halfway has it, but doesn't have it down here. So let's see. Cross them up. X, X, heterozygous. X, Y, and we have this person having it. We got X, X. Also, this one is no and no. So this person does not have it. And we get another X, Y. And this person does not have it. So let's look at those 4 results now. Is there a 25% chance that their sons will have hemophilia? Is there a 100% chance that their sons will have hemophilia? Is there a 0% chance that their daughters will have hemophilia? Or is there a 50% chance that their daughters will have hemophilia? Only one answer is correct there. The only way somebody will have it is if they have, if the XY has it, it'll look like the big letter N. If the XX has it, they have to have it on both because it's a recessive trait, so they would have to have 2 ends. So this person would have it and this person would have it, but you got to look at the 4 results. So I got 2 C's so far and an A. So let's eliminate B and D. We're good on that. It's not a 100% chance their sons are going to have it, and it's not a 50% chance their daughters will have it. So look at the results, and what do you think? I got mixed results, A's and C's. Well, let's take a look. If they have sons, this person has it, this person doesn't. So it's a 50% chance their sons will have it if they have hemophilia. So the answer is C, 0% chance that their daughters will have it, because there's nobody that looks like this. So that's a 0% chance that their daughters will have hemophilia. Okay? So again, guys, you put down the male and female first, then you fill out, big N, little n, or whatever letters you want to use for the sex-linked trait. In order for it to show up on the X chromosome, it would have to show up both times for female or just one time for male. That's why males have more hemophilia, they have more color blindness than females, because they only have to show up once, genetics. Females would have to show up twice. All right, and then for the last half of this, guys, we're going to be talking about 305, pretty much. This is how DNA, RNA, and protein are made inside of our cells, also with genetics. So first of all, difference between DNA and RNA. DNA is deoxyribose, RNA is ribose. They all have A, C, G, but DNA has T. And RNA is ACG, but RNA is U. DNA is a double helix. RNA is a single helix. DNA codes for making proteins or RNA, and RNA is just a copy of the DNA. So they're all very similar. Main thing you need to know is DNA is double-stranded, RNA is single-stranded. DNA has ACGT, RNA has ACGU. And then replication, the process used by cells to copy DNA. The enzyme unzips the DNA, and each side of the ladder acts as a template for building of the new half. I will show you this in a second. But all replication means is DNA making another copy of itself. So it's very simple. I will show you here. First step, helicase unzips the DNA. We have the dark blue, think of the dark blue as DNA. Helicase is an enzyme that unzips it. That's step 1. Step 2, then DNA polymerase, and also step 3, DNA polymerase will begin base pairing. So if there's a T here, it matches with an A. If there's a G, it matches with a C. If it's a C, it matches with a G. If it's an A, it matches with a T. That's what base pairing means. You just match the letter with the one that goes with it. So step 1, helicase unzips it. Step 2, DNA polymerase base pairs. Step 3 and 4, well, it does it in 2 different strands. There's a leading strand where it doesn't skip a beat, and there's a lagging strand where it does it in pieces. Those pieces are called Okazaki fragments. And once those pieces are made, they get glued together by ligase. So, step 1, helicase unzips it. Step 2, DNA polymerase base pairs. Step 3, it does it in 2 steps, a leading strand all in one piece, or a lagging strand in a couple different pieces. If the lagging strand has pieces, they're called Okazaki fragments. Then, those Okazaki fragments are glued together by ligase. Final product, 2 new strands of DNA that originally came from 1. That's it. That's the whole DNA replication. I can say it one more time. Step 1, helicase unzips it right here. Step 2, DNA polymerase base pairs. T to A, G to C, C to G, A to T. Leading strand is in one giant piece. Lagging strand is in pieces called Okazaki fragments. And those Okazaki fragments get glued together by ligase. Final step, you get 2 exact copies of DNA. Switching gears a little bit, that was replication. All of you, I asked on your DBA in Module 3 this question. Can you summarize transcription and translation? Every single one of you told me about this. So with transcription, think of it like part 1, and translation, part 2. In part 1, the goal is to make RNA from DNA. That's what has to happen first. You got to make RNA. And it happens very similar. Helicase unzips it. Instead of DNA polymerase, now we got RNA polymerase. And they base pair E to A to U, that's different, C to G, G to C. And then you will make the code, which is messenger RNA. That's your goal for transcription. Start with DNA, helicase unzips it, RNA polymerase base pairs now, and you make messenger RNA. Translation is the second half. Now you have messenger RNA, and that is going to be decoded or translated to build a protein. So the goal for everything in transcription and translation is to build a protein. Transcription's first. That's where you start with DNA and you make the messenger RNA code. Translation is second. That's where you use the code to translate to build a protein. Let me show you here in another picture. Here's your cell. Here's your nucleus of the cell. Transcription takes place in the nucleus. You start with DNA, and you make messenger RNA. That's transcription. Then, in translation, now the messenger RNA goes into the cytoplasm of the cell, and with the help of a ribosome, it will translate the messenger RNA 3 letters at a time. And each 3 letters stands for another amino acid. So we keep making these amino acids until it groups together to form a protein. So the whole point is transcription, where you start with DNA, you make the code, which is messenger RNA, and then step 2, translation. Now the code is going to get translated to build the protein. Transcription happens in the nucleus. Translation happens in the cytoplasm. So when you translate a code, you're going to see this table. It's going to be given to you on the biology EOC, so you do not need to memorize this. However, you need to know how to use it. So it's very easy. So you look at the first letter here, you look at the second letter here, and you look at the third letter here. So let's say I got, my first one is UAC. Which amino acid is UAC? UAC. Which one is that? UAC. So which amino acid? It's one of those crazy words you see on there. Good job, Kaylee. Yep, Catlin, that's the abbreviation for it, but you got it. Tyrosine. Look, UAC is tyrosine. That's one of the amino acids. Let's say we have a code of CCC. What's CCC? Yep, here it is. You can look this way too. CCC is proline. So let's say now I got, let's see. Don't worry about these up here. Let's go with, I'm going to type it in the box, and then you tell me the 3 amino acids that go with it. So I'm going to go this one, and GA. Okay. I just typed it in the chat box. AUG, CGU, UGA. Tell me the 3 things that go with it. So let's translate that mRNA code. I gave you an example of an mRNA code. You need to give me the 3 amino acids or the 3 things that go with it. Very close, Catlin. Just look at the last one. There you go. Good job, Paris. You think? Yep, you got it. Kaylee, you got it. Very good. Remember, you just read them 3 letters at a time. That's it. So AUG, here you go. It's your start, or methionine, your start codon. Then we got CGU, which is arginine, which is right here. CGU. And then we got the stop codon, which is UGA, right here. So just make sure you know how to read these guys and know what they translate to. This is part of translation. When you guys are reading this, a ribosome is actually the real thing that codes this. tRNA is what brings the amino acids over, but RNA is what does it. More optional videos. Again, guys, you need to know replication, the one I just explained, where DNA makes a copy of itself, and you need to know transcription and translation, because you're going to need to be able to explain that and answer questions from that on the test. You can't read off of notes, but you just need to explain it, just like I did, and you'll be fine. Okay, and here's a couple fillers. Again, most of your effort should be on what we just talked about, but here are some extra things that you might see a couple questions on in the test. There's definitely technology being done with DNA all the time. DNA extraction, that's just simply opening cells to separate and isolate DNA. There's cutting DNA, where the DNA molecules are cut into smaller fragments using restriction enzymes. And there's separating DNA as well, where DNA fragments then can be separated. So you extract it first, taking it out, opening the cells that have it. Then you cut the DNA, you take out, it's almost like you're literally cutting the pieces of DNA that you want to look at. And then you can separate DNA and include different DNA to make things better, or however you want them. So that's the 3 steps on DNA technology. Extracting it first, cutting it and selecting it second, and then separating it and using it third. And then fourth, they can sequence the DNA. That allows scientists to determine what the bases are. So once they have the DNA and separate it out, then they're going to open up that DNA and look at all the letters, A, C, G, T, and billions of letters long. And then they can cut DNA from 2 sources with the same restriction enzyme and combine them. So after they know what they have of the DNA out, then they can get pieces of other DNA and combine them together. And then they can copy them using the PCR. They can develop many copies of the same DNA. So let's say they want to, for example, they want to take out a DNA that makes bananas healthier. First of all, first step, they get the DNA extracted from the banana by doing a couple different tests. You can do this in a lab at home. Second, then they're going to cut the DNA that they think is responsible for making the banana healthier. Then they'll separate that DNA out. Then they'll look at that DNA letter by letter, base by base, to see what it is. Then they'll get that healthy gene and include it and make copies of it, and then put that into other banana DNA templates. And then they can just make bananas in a lab that are the healthiest bananas because of this DNA sequencing. So it just shows you that there's technology being done with DNA even today. Here's a question that goes with it. So here's one last ABCD multiple choice question. While genetic engineering has positive benefits, there are also concerns associated with widespread use of genetic engineering. If many farmers begin to plant more genetically modified crops that have an increased tolerance to insects, which of the following may result? So which one could happen? A, B, C, or D? If farmers begin to plant more modified crops that have a better tolerance to insects, which of the following may result? Looks like we're figuring this one out. Remember, if they're genetically modified, they're all going to be exactly the same. So if they're exactly the same, yeah, you're going to have a decrease in how diverse the crops are. Remember, with evolution, it's good to have a little bit of a diversity, because then that crop can select its own traits and become stronger. But if you give all the crops the same exact traits, you have no genetic diversity. That could be okay, but that can also lead to bad things as well, like one disease wiping out everything. So that's the only negative for genetically modified crops. Not an increase in pesticides, because that's why they made that. Not an increase in contamination, didn't talk about that. And not a decrease in crop productivity. It'll still create many crops, but the genetic diversity is the thing that gets hurt. And that's it, guys. We are all done with today. You definitely need to get out of this review thing the Mendelian genetics, remember, with homozygous and heterozygous and doing Punnett squares and interpreting Punnett squares. And you also need to know about replication, transcription, translation, and how to explain them. Do that first, and then look over those little things like incomplete dominance, complete dominance, genetically modified stuff. You can look at that last. But that's what you should have got out of today. And you should be working through the first 8 pages of your review packet. So if there's any questions you guys have, feel free to let me know. But if not, you're free to go. If you came in late, just let me know. I think I had a question earlier about tutoring for the EOC. I wasn't really sure what that was about. But if anybody needs help with anything, yes, for sure. Have anybody contact me, and I'll be happy to help. And to answer a couple good questions here, GMO foods are not bad for health as long as they modify them to be healthy, right? If they make a food healthy on purpose, then it's going to be good for you. But you want to be careful with genetically modified foods because sometimes it can eliminate all genetic diversity. And you want to have a diversity, at least naturally, so these crops can continue to grow on their own as well.